Let me introduce Heather. She's an assistant professor at Florida International University in the Marine Science Program, where she has a molecular evolution research lab and teaches invertebrate zoology and genetics. She has a broad interest in the evolution of marine invertebrates with an emphasis on crustaceans. And much of her work has been conducted in the Gulf of Mexico, Caribbean, Western Atlantic, and Eastern Pacific Ocean, where she has helped organize and participate in several major research expeditions as part of the GoMRI consortia at DEEPEND. She leads the crustacean and cephalopod genetic components. So today she will be sharing about the new and emergent molecular techniques used to study how organisms respond to stresses in the environment, as well as discussing the work of DEEPEND, the consortium as a whole. And we have a little fun fact about Heather that when she's not in the lab, field, or office, she loves participating in triathlons and almost anything outdoors related. And she thoroughly enjoys dining on her study subjects. Just the control group, she hopes. So on that note, Heather, we're going to go ahead and turn it over to you.
Great. Thank you so much. And thanks, everyone, for coming today. I'm sorry I couldn't be there in person, but I'm assuming the room is packed, although I can't see you. But again, I just wanted to thank you for the invitation. And today I'm going to be talking about what my lab is specifically in charge of. And the title of my talk is Traditional and Emergent Molecular Techniques to Characterize Species Biodiversity and Ecosystem Health. And before I get started, I do want to acknowledge that I am just one piece of a very large team. I am part of a consortium named DEEPEND, and this consortium is led by Dr. Tracy Sutton at Nova Southeastern University. And here's just a few names of the very talented collaborators, research scientists from multiple university and institutions around the United States. And of course, all of this work would not be made possible without funding from the Gulf of Mexico Research Initiative. So let's get started. So really, the mission statement of our consortium is really we want to characterize the oceanic ecosystems of the northern Gulf of Mexico. And we're doing this because we're trying to infer baseline conditions in the water column. With this information, we hope to establish a time series with which we can detect natural and anthropogenic changes that happen within these ecosystems. So one way that we are going to reach that goal, one of our major objectives of the DEEPEND team is to really characterize biodiversity. And the questions we're asking are questions like, how does biodiversity vary with space and time? In order to do this, we need to go out and sample that biodiversity. So what we do is we have 2 research cruises every year. We've completed, this project started in 2015. We've completed 4 two-week-long research cruises. We go out and we sample the communities that live in the water column. And we do this off of the research vessel called the Point Sur. And I just wanted to introduce you to the nets that we use to collect these organisms while at sea. The net we use is called a 10-meter squared mock net, and that's what you're looking at a picture of right now. This net is actually made up of 6 individual nets. These nets can be opened and closed at very discrete sampling locations or localities. So we're opening and closing these at very discrete depth intervals so that we get a snapshot of what organisms are living at these different depths. So it really gives us a good idea of the diversity and the abundance at different depths as we move down the water column. We do sample all the way from the surface up to about 1,500 meters. And sometimes we've even gone as deep as about 2,000 meters, which is about equivalent to 6,000 feet. So this is how we sample the water column. We bring these organisms on board, and we're really interested. There's 3 major taxon groups that the DEEPEND consortium is interested in. We have a team of fish taxonomists. This team, we sort the samples on board, we identify them. So we're interested in looking at fish taxonomy. The other two groups that we're interested in are crustaceans. This is the group in which I'm a part of. I'm part of the crustacean taxonomist team, and we also have a cephalopod taxonomy team on the ship. So another method we use to characterize the biodiversity is genetic techniques. And so we're using genetic techniques to really document the biodiversity that we're finding and also to create species inventory. And this is really what I'm going to talk about for the rest of today. My group specifically is in charge of the crustacean genetics, and I'm going to talk to you a little bit about the methods that we use to characterize biodiversity using genetic techniques. So the technique we use is called DNA barcoding, and really this is a method that we use to identify organisms. This DNA barcoding is really based on the premise that every single organism has a unique DNA sequence for each species. Okay, so basically we can sequence very specific regions of an organism or a species' DNA, and that DNA is going to be specific. The code in which that DNA holds is going to be specific for that species. So much like assigning a numerical barcode to a shopping item, we can assign a DNA barcode for each species that we find. So here's an example of what DNA barcoding looks like. Here you're looking at 6 different species of fish. Each of the colors represent a different nucleotide or DNA base pair. So blue represents, we have blue, red, green, and black representing A, T, G, and C. And you can see there's parts of the DNA where all 6 species have the same DNA sequence, but you see that there is variability, and it's this variability in that DNA sequence which allows us to identify those to species. So how do we do this? So how do we do DNA barcoding? We basically extract the DNA. Typically for crustaceans, we're extracting from muscle tissue. We use something called the polymerase chain reaction. This is the way that we amplify that piece of DNA. We sequence that DNA, and we have to be careful of what genes that we're selecting. So I told you that this is all based on variability. So we need to select genes that have variability in their DNA sequences. To date, we've barcoded, just from our 4 research cruises that we've gone on, we've barcoded over 481 species of fish. It's an extremely diverse habitat for fish, 100 species of crustaceans, and about 20 species of cephalopods. So really why? Why are we using genetic techniques to characterize biodiversity? Well, this really allows us to identify organisms based on a DNA sequence. And why would we ever need to do this? This turns out to be extremely useful when we are trying to discover new species. You can imagine that if we bring up, a lot of the specimens we bring up in these nets are sometimes damaged, mutilated, or we're unable to identify them just based on traditional taxonomic methods. So this is where we can actually just take a very small clip of tissue, sequence the DNA, and we are able to identify what that organism is. Of course, DNA barcoding is it's dependent on having some kind of database with all of these sequences so that we know we can positively link whatever we find to a database of known species sequences. So we're starting to create these species inventories especially for the deep sea where a lot of this has never been sampled before. You can also imagine that this is really helpful. It allows us to make large linkages. So what do I mean by that? So a lot of times larvae look extremely different from their adult counterparts. Here's an example of a paper that I published back in 2012. So this larval form here was called Ceratapis monstrosa. It was found only in a few instances. No one knew what this was a larval form of. Actually, no one knew what this was in general. but we were able to use DNA barcoding to positively identify it. So this is just a larval form of an adult shrimp. This adult shrimp is really it lives in the very deep sea up to about 5,000 meters. So DNA barcoding can also allow us to make these really larval adult linkages. So I'm going to shift gears a little bit and start talking. I'm going to move away from barcoding and get into ecosystem health, recovery, and resilience. So the next thing that all of us in DEEPEND are really interested in are really addressing these long-term consequences of the Deepwater Horizon spill on pelagic communities. So Monica, introduced the plume that was present after the Deepwater spill. So really, we have to remember that this spill was really a deepwater event. The wellhead leaked at about 1,500 meters. There was a massive plume that developed at about 1,000 meters. So there's been a lot of research on the effects of surface organisms, but our group is really interested in what is the effect of that huge deepwater plume, subsurface plume, on these deeper pelagic communities. So again, one way that we can study these systems is to use genetic information. And genetic information, it turns out, is a really great proxy to measure health, resilience, and recovery of an ecosystem. So what metrics am I actually talking about? There are two metrics that we're measuring, and one of these is genetic diversity. Genetic diversity turns out to be a really great proxy for the health of a population, which in turn can give you information about the overall health of an ecosystem. So genetic diversity is exactly what it sounds like. It's the amount of genetic variability within populations of a species. And this chart just diagrams the really important effect genetic diversity can have on the ecology of an organism. So the black lines represent direct effects. The dotted lines are indirect effects of genetic diversity. So it has an effect on the population fitness, performance of organisms, the diversity and abundance across multiple trophic levels, which eventually trickles into whole ecosystem properties and community properties. So we're using genetic diversity as a proxy for ecosystem health. The other metric we're using is something called genetic connectivity, and this serves as a proxy for ecosystem resilience and recovery. So genetic connectivity in the simplest sense is really just how connected are those populations? How much genetic information are they able to share? Is one population in an area able to share with another? So this has to do with the exchange of genetic information. Are they breeding? Are they able to share genes or genetic information with neighboring populations? And what you're looking at is a map of Spain. When you see the thicker red lines, or just the thicker lines in general, are populations that share a lot of genetic information. This is called gene flow. So it has very high gene flow between populations, where the thinner lines are populations that seem to be reproductive and genetically isolated from one another. So you're looking at one species. This is one species of mullet that was collected along the entire coast of Spain. So this is basically showing you that there's a lot of genetic structure within this one species. There's regions where these populations can interbreed and share genetic information, and there's other regions where they cannot. You can imagine that genetic connectivity is really important where you might have some kind of event that wipes out an entire population. If you have neighboring populations that can genetically rescue or contribute back to that population, this really plays a role in the recovery of the populations at that specific time. So how do we measure genetic diversity and connectivity? we are looking at, this is a population level measurement, so we are sampling different populations within multiple species across time and space. We're extracting DNA from about 20 to 30 individuals per population. There's been a lot of recent advancements in DNA sequencing, and our team is really using the gold standard for how we measure genetic diversity and genetic connectivity. This method is a very long name, so I'm just going to refer to it. It's called RADseq. And RADseq really allows us to use the entire genetic information of an organism to look for regions of variability. So it allows us to study diversity and connectivity at very fine scale, at a very fine scale. Okay, very quickly, how do we do this? So this is the protocol that we're using in my lab. So you basically, you take an individual, you pluck the tissue, you extract DNA. The very first step is you take that DNA and you shear it into multiple very small fragments. You shear it with something called restriction enzymes. These enzymes cut at specific points in the genome of an organism so that you know exactly where it's cutting. This results in a bunch of different DNA fragments. So at this point, your individual, the DNA is sheared into a bunch of different fragments. The next thing you need to do is you need to be able to link those fragments back to the individual. So we attach something called a barcode. This is different from the barcode I talked about previously. This is just a very short segment of DNA that allows you to identify those pieces of DNA back to the individual. You pool all of these together. You do this multiple times across a bunch of different individuals. And then we sequence all of that DNA on really fancy machines. And these fancy machines are called next-generation sequencing platforms. They allow you to sequence millions to hundreds of millions of base pairs of DNA. So we have a lot, I mean, we're talking about a lot of information you're getting. So we use bioinformatic tools to sort back out those DNA sequences, and we're allowed to, and then we can make estimates of genetic diversity and connectivity. So quickly, we are comparing genetic diversity across both time and space. We've sampled from 2010 and 11, so we have samples before and right after the oil spill. We're looking at changes in genetic diversity till now. We're also looking at connectivity. We want to know if populations in the Gulf of Mexico were wiped out. Can those be genetically rescued from populations, say, in the North Atlantic? Okay, and the last minute, I'm going to talk about just a few other of the field sampling technologies that my consortium uses. Not me personally, but I do think it's really interesting. We have several cruises, 6, led by our colleagues at Texas A&M that are out sampling the plankton communities. This is just a graphic of some of the areas that they're sampling the plankton. We have bioacoustics, so we have 2 researchers that are leading the bioacoustics part of this grant. This is where we're using sonar, and that sonar allows us to measure particles in the water. Those particles are typically organisms, and how they move through the water column. So we can measure how biomass is, using sonar to see how biomass is moving through the water column. Of course, we're taking as much oceanographic data as possible, salinity, oxygen. We're interested in currents. We're measuring where that loop current is coming up into the northern Gulf. And we're able to partner with the Naval Research Team to model a lot of this. So before we actually go out on our research cruise, we use these models to predict where eddies are going to be forming. Because remember, we're really interested in the distribution of biodiversity, and we're looking for major oceanographic features like eddies, like the loop current, and we want to sample within some of those features. We are also we're using that and sending out these things called gliders. We're using satellite imagery to really ground truth all of this, to bring it all together, to have a really integrative look at the oceanographic biodiversity and ecosystem health. And with that, I thank you. I encourage you to visit our consortium website, and I'll take a question if there's time. So thank you very much.
Thank you, Heather. Anybody have any questions for Heather? None? Yes, Brett, go ahead. I'll relay. If you can speak loudly, that'd be great. And I'll ask if she can hear you. Go ahead.
Well, I'm just wondering if level that genetic diversity and the genetic connectivity findings are related to deepwater horizon versus, other.
Heather, were you able to hear that?
Can you repeat the question, please?
He's asking what is the confidence level of your, what was it, the two
differences in genetic diversity and genetic.
The differences in genetic diversity and genetic conductivity as far as directly from the Deepwater Horizon or other spills that have occurred?
Yeah, absolutely. So, so you're absolutely right, and we have to be very careful with the interpretation of the findings. So the very first thing we want to do is just to get baseline data. So we want to know what the genetic diversity is within these species that we've selected. And really, we're looking for major shifts. For example, we're looking for major shifts in genetic diversity from, say, our pre-spill or directly following the spill to maybe 5 to 7 years later. And of course, we can't make we won't be able to make a direct link, but we are looking for shifts and seeing if we see changes in genetic diversity. We are submitting all of the samples that we're using in these studies are undergoing analyses where we're measuring PAHs so we can know if those specimens have been directly exposed to oil. But again, you have to be careful. We don't want to be overconfident in our results of saying that they are directly linked to the oil spill. In terms of connectivity, I think that this is going to allow us to not necessarily relate it to Deepwater Horizon, but it will allow us to have confidence whether those populations in the Gulf, if something is to happen, if we are to wipe out a population or an entire population or severely impact the genetic diversity so that it's lower, are they able to be genetically rescued from populations that are outside the Gulf of Mexico? So this is the type of information we're trying to gather.
All right, thank you. Yeah? Oh, sorry. Go ahead.
Yeah, my question is, all of the populations in the Gulf connected, or are there differences in connectivity among the different populations or different regions that you sample?
Yeah, absolutely. That's an awesome question. So, I mean, that's another thing we're definitely integrating into the experimental design. So I wasn't able, is my screen still shared, Monica?
Yes, it is. Mm-hmm.
Okay. So this is basically some of our sampling stations. We are not sampling all of these. These are all of the sampling stations that were sampled back in 2010, 2011. We are not able to go back to all of them, but you can also see that we're not sampling a huge region, right? It's restricted to the northern Gulf of Mexico. So to answer the question of connectivity across the Gulf of Mexico, it's going to be very difficult. To answer that question, we would need to sample definitely regions of the Gulf, the different east, west, south regions of the Gulf to see how connected the populations in the Gulf are. We are trying to set up our experimental design to include stations or samples from stations that are as far apart as possible to see, do we see any population structure within the Gulf? I think the really interesting question is, we are sampling inside and out of the loop current, and this loop current is bringing up organisms from the Caribbean, right, up through the channel. So one of the questions we are asking is, do we see is there population structure inside and outside of that loop current? So that is one thing we're integrating into the experimental design.
Yes.
Yeah, Heather say that they did sampling before Deepwater Horizon.
So we have some samples that we're going through now that were in early 2010, directly right after the spill from 2010 and 2011.
I'm not clear. Was that before or after?
Was that before or after the spill? Do you have any?
We have some samples from before the spill for groups like the fish, and we have other samples from directly following the spill. Now, if we're using samples directly following the spill, there's not been enough time for those samples to affect the genetic diversity. We're also comparing it to, we kind of have a control, which is we have samples from outside of the Gulf in the northern Atlantic to also compare to. So we have both.
Any other questions? Oh, thank you again, Heather, for participating.